for the stakeholders in vaccine development and implementation to be at the table together at an early stage, and stands us in good stead to think about the up-and-coming initiatives for vaccine manufacturing on the continent. So anyone who wants a deep dive into what was achieved during this time, there's a supplemental issue in CID which really So Menafrivac was introduced then after licensure and WHO policy endorsement in a very staggered way, because obviously there wasn't vaccine available for everybody also at the same time. And various catch-up campaigns were run before the vaccine was rolled out as part of the EPI program. And when you look at this, by 2016, 2017, millions of people had been vaccinated with this product, which had come through, still, took 8 to 10 years to come to fruition, but the entire belt was eventually covered, and this vaccine is now part of our immunization schedule also here in the Gambia. Not only was safety and efficacy shown, but very, very quickly effectiveness was also demonstrated here in data of the country, but not in the other. And you can see the flattening of the curve there immediately after introduction of the vaccine. There was also sustained effects. Of course, the job is never quite done, because not only do we have MenA, and Jolene yesterday alluded to the evolution of other strains of meningococcal disease also in the area. So we're now looking at a pentavalent vaccine, and again, 2 sites in the region have stepped up to conduct these trials, and I'm sure Dr. Clark will give you more information about the outcome. But there is very good head-to-head comparison with the existing conjugated, very expensive vaccine from another company, and it is expected that this vaccine will also be used for outbreak responses and will then deal with the remaining meningococcal problems that we have in the belt. But all things are not equal, and global access and vaccination coverage is something we need to look at very critically. Because here in the data from the excellent website Our World in Data, we can see that vaccine licensure since the introduction of EPI has clearly, the EPI program has made enormous strides, and you can see here these trajectories after introduction of vaccines in our EPI plans, but you can also see that things have started to plateau around the 80%, and some vaccines here, for example, are already looking as if they're plateauing, although that might change with the new pneumococcal vaccine. And what is also really unacceptable, that it has taken such a long time between having vaccines that were already proven to be safe and effective in high-income countries to come to the continent. And 10 years of delay between these sorts of interventions reaching Africa, I think, are no longer acceptable. And I believe that since the SARS-CoV-2 pandemic, this has really risen to the forefront of our thinking. So, even when we have the vaccines available, the access to those vaccines is not always easy, and the implementation challenges remain. And Dr. Wariri presented a couple of areas of research that we are exploring in trying to go into more depth what is really underlying the dropout and the equity gaps in immunization systems in West Africa. And here's just an illustration of the quite impressive trends in dropouts from the first dose of diphtheria, pertussis, and tetanus contained. And in many countries, the GVAP dropout targets across West Africa are still not achieved, and we need to really look at what we need to look at. And you can see that things have improved dramatically, but they're not actually improving further. This is a line of dates in years. And again, there are problems that are quite distinct in particular countries, and without wanting to single out particular countries, the number of people who live in Nigeria who have not received the overall DPT-3 cover is really a large number of children in this regional area. So there are only a few countries, and I was very pleased to see the Gambia be made here, and this is just EPI and DPT-3. Similar things can be said for measles progress, and here's the scorecard that the team devised for looking at measles coverage for just the first dose of the measles across the dark green areas, but again, there are large areas where elimination of measles is way off. And when we're looking at the second dose of measles coverage, it is even less so. So a lot more effort across the 15 ECOWAS countries need to be made to make sure that measles remain under control. And even in the Gambia, after the COVID pandemic, we have seen measles outbreaks, and there's been a campaign recently. Moving on to vaccine confidence, because many people think the reason why we're seeing low coverage is because people don't want to take the vaccines. This is a piece of work from the Vaccine Confidence Project from a few years ago, but the map doesn't look that different now, where questions were asked, do you agree with the following statement, vaccines are safe? And you can see anyone in orange and red did not think that this was the case, but reassuringly, there are many green patches on the African continent. And I have actually more confidence in the vaccine acceptancy on our continent than I have in Europe. And the conversations we are having with people on the ground, which have also extended to quite extensive focus group work and social science work carried by one of our PhD students who also presented to you on Monday afternoon, is that there are, of course, contextual influences, there are individual and group influences and vaccine and vaccination specific issues, and they do play into people's confidence. But overall, the majority of people on this continent are only too delighted to receive vaccines for their diseases that they still perceive as a personal health risk. And when we explored this in the context of COVID vaccines, people kept saying, why didn't you bring us a malaria vaccine? Because this is a disease that we really feel on a daily basis, whilst COVID was a disease that people didn't experience in the same way in the countries here. So we need to always ask ourselves, are we making the vaccines for the people? I think the rollout of the SARS-CoV-2 vaccines has been an excellent example about some of the tensions in this space. And I might be making very incorrect, politically incorrect statements, but I was surprised to see that the African Union Bureau of Heads of States endorsed a strategy of setting a target of having at least 60% of the population vaccinated with COVID, SARS-CoV-2 as it was in high-income countries. So what was driving this agenda? And was this an agenda that was fit for the needs of the region? And I'm not here to say what I think. It's about us to discuss within the region what we think. But the tracking on the uptake and the trajectory of these promises can be academically assessed. And here's a bit of work from Anne at the London School, who showed, given the trajectory that was actually visible with the COVID vaccine output, the sub-region would have had less than 1.6% of the total population fully vaccinated after 18 months of vaccine deployment. And the speed of vaccination would have had to be increased 10 times, 7 times, and 4 times to achieve 9, 12, and 8 months of vaccine deployment. And I think we really need to ask ourselves whether the target that was set was really a target that was needed here, because clearly it was all hands on deck, but it was the same people doing all the work and the same people who were driving COVID vaccine rollout compared to. If we look at this paper from on to routine childhood immunizations during the COVID-19 pandemic, it is obvious that the deaths averted by vaccination with the standard EPI vaccines were a lot higher than the deaths who would have been averted by COVID-19 vaccines. And there was a benefit-risk ratio calculated here in this paper. And just to pick out measles, for example, or yellow fever, there are hundreds of thousands of cases that children were potentially exposed to, could have died of, whilst there were only a few hundred that they might have succumbed to COVID-19. And to me, the children were really losing out during the COVID-19 pandemic, both in terms of healthcare delivery. We have seen a slight blip in infant mortality through our HDSS data that Anna showed only yesterday. And this is about access to care. This is about health system strengthening. Vaccines are just an indicator and not necessarily the cause. However, there have also been amazing silver linings from the COVID experience. Vaccines can be developed at speed, clinical trials can be done lots more efficiently, the regulatory procedures have been streamlined in a way that we've never seen before. And we have seen that good community communication is absolutely essential for implementing a vaccine program, whether you're in a high-income or a low-income setting. And equity of access become an international focus, which can only be a good thing. And self-sustainability is a priority that has now resulted from a lot of the momentum that we have seen around the COVID rollout, with vaccine manufacturing now being a concept that is being embraced through the African Union and the Africa CDC, where I feel that institutions like our unit can also make a significant contribution to Africa investigator-led studies, improve clinical trials capability, and also technical developments, and this goes closely with diagnostics, which I alluded to yesterday. So if we're looking at where the COVID vaccines came from, it is obvious that there was a very, very small percentage of vaccines that were actually made in Africa. South Africa had, a very small role to play here, and we know that it's the majority, I think about 90% of our vaccines at the moment come from outside the continent. And this prompted the Partnership for African Vaccine Manufacturing and set a framework for action, which is really a document I would urge you all to have a very good look at, because I have found a lot of really detailed descriptions about how this process could now move forward. And it's a shame that, so the goal is to produce and supply 60% of the total vaccine doses required by the continent by 2040. And there are several framework of action programs here that relate to market design and demand intelligence, access to finance, obviously a huge issue, regulatory strengthening, tech transfer, and where I feel the institutions in the academic setting in the continent can play an enormous role, including ourselves, is the R&D, talent development and sustainability. Regulatory processes will require harmonization, and initiatives that were started 10-15 years ago on the continent are now stepping up to identify what needs to be done next, to develop a regulatory framework that actually allows regulatory approval of vaccines that are also made on the continent. This is all still in very close collaboration with the WHO, which it should be, and the national regulatory authorities have been given milestones to achieve that. We have regulatory authorities at a level that's called the ML3 level, where vaccine excellence and regulatory procedures can be confirmed, and this all builds on existing initiatives. And I think the continent is coming together. It's greatly also thanks to the African Union and the leadership from John Kangasong in this space to really build up a potential for a thriving vaccine manufacturing industry on the continent. There is a timeline and there is a strategic plan and operationalization, and the good thing is you can see it's already happening. And when we look at the vaccine manufacturing before 2020 here, there were a couple of vaccines being mainly fill and finish in South Africa and in Egypt, and now we have various initiatives, and I've just pointed out here the West African one at the Institut Pasteur, but there are also substantial investments in Rwanda and Nigeria. And I think industry is coming to the table, partially due to international pressure that is no longer acceptable to just keep the IP and the technology in-house. Coming back to where I think the academic institutions will play a leading role is on the disease prioritization, because as you can see, for many diseases that we have in our areas which might be prone to outbreaks, there are currently no vaccines. And if we want to demonstrate the need for these vaccines and also ultimately their effectiveness, we do need to get on the case and get the surveillance data generated that will actually support the case. And that relates to sentinel site surveillance, and there's a program here in our new QQ to have surveillance programs in place for places where people present with fever, and I very much look forward for that being upscaled. And there will be many, many diseases that we might not have covered with our diagnostics at the moment because we're very focused. But we now have multiplexing diagnostic platforms, and we can make a contribution to inform the priority setting for the framework of action, which is driven by the needs of the countries and the geographical areas in which we find ourselves. So of course the elephant in the room is the funding, but this is also not completely hanging in the air. About $30 billion will be needed over the, but obviously to mobilize the funds for these investments is not going to be easy. And actually R&D is taking a large amount of the chunk. And I've put here 2 stars because I think this is where academic units like ours and others in Africa can make a contribution through our science being made available for the public health good in combination with the needs of trying to define what the next steps are in R&D and setting the agenda. No talk would be complete without alluding to climate change and its health impact. And the team yesterday did a wonderful job, despite of all the technical issues, to really make us understand that there is a crisis, and I've just pulled out a few bits and pieces where I think that vaccines will be able to make a contribution, and we need to look at that. So this is the 2022 report of the Lancet Countdown, which appeared just over a couple of weeks ago. And without going into too much detail, all you need to really perceive is all the curves are going up. And here is the distribution of dengue. Here is Vibrio cholera, and the conduciveness of the environment for carrying these pathogens cannot be underestimated. And even the BBC headlines have finally picked up that there's a cholera outbreak in Lebanon. Cholera was mentioned yesterday as sort of bits of epidemic that pitch up also here in Africa, mainly imported. But that there's a cholera vaccine shortage. And this is a global issue at the moment, and it's the reason for it is that one company has basically downscaled their production. They announced this a long time ago, but no contingency plans were put in place in time. So we now have a cholera vaccine shortage, which is turbocharged by climate change. And, we need to ask ourselves, are we in a position where we can contribute to the vaccines that are needed for such emergencies? So, in my view, there's no meaningful conversation to be had about any urgent health challenges now without acknowledging the role of climate change. And this is a picture I took when Andy Haynes gave a similar talk to what he presented yesterday at the Gates Global Challenge meeting in Brussels. And I've just pulled out from this report the areas where it relates to waterborne and vector-borne infectious diseases. And you can see that there's a whole number of infectious diseases that we are already also working on, doing surveillance, could possibly do more, especially here in the endemic viruses, where also really increased outbreaks would come from and where new vaccines are desperately needed. So there's a genuine opportunity for African research in this space, in my view. And we must also not forget about the impact of conflict and migration as enemies of vaccine coverage. And this relates to cholera, as we've just seen, polio. The only countries that still have endemic polio are countries that are also having ongoing conflict. Measles, Ebola is not going to make our life easier in conflict-ridden countries. By which time I'm not meaning Uganda as a conflict-ridden country. It just so I think the immunization agenda remains unfinished, but I also think it needs to now go beyond the valleys of death. And if I had to write this paper again, I think I would very much extend it to thinking more about the implementation challenges. And if I want to bring this all together, what could Africa-based research institutions do next to contribute to vaccine research and implementation on the continent? And I hope I have given you some incentive to think about the collaborations with the African Vaccine Development Agenda, with the work in the context of World Immunization Agenda, which no doubt Dr. Clark will shed more light on, but in particular to drive R&D partnership in collaboration with the public health institutions, and we saw that nice pyramid of how that's projected by the WHO. We also need to contribute to develop African vaccinologists with a future on the continent. And this relates to opportunities in universities and also in industry that keep the people that are trained and talented on the ground. And we need, in all we do, in my view, consider the climate change on health with a special view on mitigation strategies based on vaccines, because we need to think about how our environment will change, to which we can hopefully contribute some mitigation strategies. Of course, none of this could be done without the fabulous teams in field labs, data, clinic, transport, to name just a few. and also to emphasize that there is sustained financial support necessary. These things don't fall from the sky. The Oxford vaccine wasn't made overnight because they had the genome. It was a result of a continued investment in research and development at a key university where finally the fruits of labor that were dedicated actually to a MRC vaccine came to fruition in the context of the SARS-CoV-2 vaccines. And, you know, we also couldn't do the job without the national and international public health institutions and ministries. And I'm really proud of the 75 years of collaboration that the MRC unit has had here. And we heard from the ministry as well how they see that. It's not always easy, as everybody knows, but there's no alternative to push on. And I think in conclusion, what we can say, that the future is bright for vaccinology and vaccinologists in Africa. Vaccines work, we know that. Vaccinations work best. So let's get them to work for everyone, everywhere. Thank you.
Thank you very much. Who is a clinician, scientist, and consultant in pediatric infectious disease and immunology. And he joined the MRC unit in 2012 and has been based full-time in the Gambia since ever since.
Okay. Great. So thank you to the organisers, thank you to Beata. I'll be picking up on some of those themes and also some of the things you've heard already over the last couple of days, but really to try and give an overview of some of the clinical vaccine trials that have been done, but prioritising really things that are ongoing at the unit at the moment, to give you a flavour. So since the birth of the expanded programme on immunisation in 1974, the Gambia has really been punching above its weight, so to speak, in terms of vaccine-related research output. So you can see on this table, the Gambia was third only to Nigeria and South Africa in terms of publication output, and if you adjust then for population or gross domestic product, you can see, as you would perhaps predict, it's at the top of the tree. And in this figure, you can see it's a notable outlier on the continent of sub-Saharan Africa. But I'm sure I don't need to convince you, particularly after what we've heard over the last few days, that this is certainly not just about quantity of impact, but also, of course, quality and ultimately, most importantly, impact on disease endpoints. And it's really important to emphasise right up front, I think, that none of this work will be possible without our ongoing interaction with the Ministry of Health, not only in the day-to-day support very actively that they give us with all the vaccine trials that we do, but also, of course, with vaccine implementation, without which the results of the trials would essentially be meaningless. So I'm not going to be able to pick up on the entire legacy, and much of this has been mentioned already, so I don't want to repeat too much of the trials that have been done over the last 6 decades. But essentially, as you heard, the first trials in the Gambia, the first vaccine trials in the Gambia were for trachoma in the 1960s. And then perhaps a less well-known fact from the Gambia, apparently the Gambia was the first country globally to introduce measles transmission through vaccination. So this interesting reference from a WHO meeting in 1970, most noteworthy has been the virtual eradication of measles from the Gambia since mid-1968. Of course, that was relatively short-lived, unfortunately, for 3 or 4 years, and the unit went on to do a number of key early trials with subsequent trials with measles vaccine scheduling and also OPV trials, which I'll come back to a little bit later. No talk, of course, about trials in the Gambia would be complete without mention of the Gambia Hepatitis Intervention Study, which achieved many things, of course, but including most notably the early introduction of hepatitis B vaccines in the Gambia into the EPI schedule. The Gambia has also had a really notable history of malaria vaccine research, which no doubt Annette will pick up on in the malaria section later today. But the first trials, as we've already heard, the first field trials of RTSS, the first efficacy and the first paediatric trials were done here. And of course, RTSS has gone on and become the first vaccine for malaria to be licensed. We've also been very closely involved as a unit, and again, Annette will pick up on this later, in the DNA or vectored vaccine studies led by the team from Oxford, Adrian Hill and colleagues. So again, the first field trials, the first efficacy trials, and the first trials of young infants. And again, this is a very program that there's a lot of optimism about. And lastly, just to touch on the bacterial vaccines, which we've already heard about, the Hib vaccine, the Haemophilus influenza type B, and the pneumococcal conjugate vaccines. Perhaps most notably, I think this has already been said, but just to emphasize it, the first data on the efficacy of Hib vaccines in sub-Saharan Africa was generated at the unit. So this was efficacy against radiologically confirmed pneumonia as well as meningitis. And this was at a point, I think, when the burden of Hib disease, at least as a cause of pneumonia, wasn't completely clear. So although this wasn't designed as a vaccine probe study, it also gave important information about the burden of Hib disease as a cause of pneumonia in this setting. And of course, the Gambia then introduced the Hib vaccine first on the continent. One of the two key trials of the pneumococcal conjugate vaccines were done here, and demonstrating, amongst other things, an impact on all-cause mortality, really emphasising the burden of pneumococcal disease in this setting. And no doubt, again, Grant Mackenzie will pick up on this later. And the country introduced PCV second. Of course, these introductions are critical, but have also allowed the unit then to go on and generate not just efficacy data in trials, but also effectiveness data and impact data on both these vaccines, which Grant will no doubt expand on. And lastly, just the Menafrivac vaccine. So the key licensure trial of this vaccine was done here, and we've seen this figure several times, and Beata just presented it. It's an incredibly good vaccine, and has really removed MEN-A disease as a problem on the continent. So this is genuinely an incredible legacy of vaccine trials. It's a unique legacy, and anyone with any passing interest in vaccinology cannot help to sort of appreciate this. But I just want to now move on to think about some of the current trials that we're doing, really trying to put them in that context, because we're really trying to build on that legacy at the unit at the moment, or continue to build on that legacy at the moment. But first, before moving on to that, I just want to briefly touch on the immunisation agenda, which Beata mentioned already, because I think it feeds into many of the studies or the vaccine trials that we're doing. So the immunisation agenda really has vaccine equity, already a point made by Beata, at its heart. So it's described as a global strategy to leave no one behind. There are a number of impact goals. So first of all, to prevent disease, to reduce by 50 million vaccine preventable diseases over the course of the decade, to eradicate polio and drive the elimination of measles, and to decrease disease outbreaks, including, of course, meningitis outbreaks. It also aims to promote equity through reducing zero-dose completely unvaccinated children and driving 500 programme introductions globally at a country level over the decade, as well as to build stronger programmes both for life course immunisation and also to integrate EPI programmes, so vaccination programmes, into universal health coverage more broadly. So this is the picture of the trials that are ongoing at the unit at the moment. So we're doing a considerable number of trials are active or winding down or winding up or in recruitment at the moment. Grant Mackenzie will pick up on the pneumococcal vaccine scheduling study that he's leading in Basse later today. Beata has already touched briefly on maternal vaccination, and Kirsty Lador in the subsequent session no doubt will pick up on that in the context of sub-Saharan Africa more broadly, and Annette Earhart will discuss the malaria trial. So I just want to touch on 4 areas, really to try to give a sense of the breadth of vaccine trial research that's ongoing at the unit at the moment. So first of all, the need for a new vaccine for epidemic meningitis elimination. We've already heard, of course, about Menafrivac. So as we know, the meningitis belt stretches from Senegal and Gambia in West Africa to Ethiopia in East Africa, and has these epidemics of bacterial meningitis, largely MEN-A historically, as we've already heard. And the global roadmap for defeating meningitis, which I know Beata and others were involved in, has as its first goal the elimination of bacterial meningitis epidemics by 2030, so over the course of the decade. And we've seen this is data from 9 countries, so Menafrivac has had an incredible impact. I mean, it's just a remarkable vaccine. but this has been associated with a persistent, if not increasing, rates of non-A meningococcal disease, as well as, of course, other bacterial causes of meningitis, Streptococcus pneumoniae, Group B Streptococcus. Again, I'm sure Kirsty will pick up on that topic. So if we just look at the non-A meningococcal disease, first of all, so just focusing on that, as many of you will be aware, many of you may well have been involved, the unit was involved, there have been really serious outbreaks of Men C disease in Nigeria and Niger, ongoing epidemics of Men W disease, the emergence of Men X disease as a cause of epidemic meningitis. So it's clear that the story is not complete, and that an affordable vaccine to comprehensively prevent meningococcal epidemic disease in the meningitis belt is essential if the goals of the roadmap are to be achieved. So Beata's mentioned this, so we've been recently completed a study, which I'll show you the results of a Men-A-C-YWX conjugate vaccine. So this is the first pentavalent meningococcal vaccine that's available, and it's also the first vaccine that's expected to provide protection against men serogroup X. So it's manufactured again by the Serum Institute of India, and conducted with the Centre for Vaccine Development in Mali, so with Professor Sambasau, who is surely a titan of vaccinology in this part of the world. We recruited 1,800 2 to 29-year-old participants, and this was a non-inferiority trial led by Dr. Umesi, who you can see in the picture and at the front. So just to give you the sort of headline results, essentially, so whether you looked at differences in seroresponse rates on the left, or geometric mean titer ratios on the right, you can see the point estimates for all serogroups favour the new, so the pentavalent vaccine in both cases, and all the lower limits of the confidence intervals that we're using to test for non-inferiority are way above the non-inferiority margin that you can see. So essentially non-inferiority was demonstrated for all serogroups based on both seroresponse rates and geometric mean titers. So it's expected that this new vaccine, this pentavalent vaccine, will be licensed over the coming few months and pre-qualified subsequently by the WHO, and is likely to play a key role in the elimination of epidemic meningitis in this setting over the coming years. Now moving on to polio eradication and some of the barriers to polio eradication. So eradication's really been, we've almost been there for quite some time, but this map shows in the green dots cases of acute flaccid paralysis related to the type 2 vaccine-derived polio over the last 12 months to the 1st of November. So this is very current data off the website. And there's also been vaccine-derived polio, as you know, detected in New York and London over the same time frame. So this really is a global issue related to travel, of course. So vaccine-derived polio occurs when the Sabin, the attenuated Sabin OPV, loses some of those attenuating mutations, so it regains the capacity for transmission in the population, and also regains neurovirulence, hence the acute flaccid paralysis. So broadly speaking, there are two responses to this. IPV, the inactivated poliovirus injection, protects the individual, so you're vaccinated, you generate high levels of serum neutralizing antibody titers, and you're protected from paralysis, but it doesn't generate mucosal immunity, so it doesn't stop the virus circulating in the population, so you can't use it in isolation for control of outbreaks. In contrast, OPV is given orally, obviously, and it does therefore generate mucosal immunity and does block transmission. However, of course, OPV is the source of the vaccine-derived polio in the first place, and what has clearly occurred is in areas of low population immunity overall, the OPV has been given, but essentially fed further outbreaks. So there's been a rapid increase in cases of vaccine-derived polio associated with OPV use. So this has become, or became over 2018-19, a really major problem, particularly in the sub-region. So it's clear dose-sparing strategies for IPV. So, for example, you can vaccinate hundreds of thousands of children and protect them from paralysis rapidly, but also a more genetically stable form of OPV to prevent the problem in the first place are going to be key to Garen, who might be here. So we and others have shown that fractional 0.1 ml doses of the inactivated poliovirus vaccine, IPV, when given by the intradermal route, are dose-sparing. So essentially, the immune response you generate from a 0.1 mil dose given cutaneously, and this obviously allows one vaccine to be given to 5 children rather than just one child. However, there's real concerns if you're wanting to do an outbreak response campaign about the practicality of giving large-scale intradermal injections, given the sort of technical difficulties in giving good quality intradermal injections. So this has really been inhibitory to countries doing outbreak response campaigns with fractional doses. So we did a sort of pragmatic trial, really working very closely with the government of the Gambia and trying to emulate the processes that they use during an outbreak response. So we compared 3 devices, including this needle-free jet injection device, which you can see this girl tolerating very nicely. We compared injections, so the BCG injection, with inexperienced nurses who'd really never given a vaccine intradermally before or had very little experience of it. And over the course of this sort of campaign, we vaccinated 3,000 children over a course of 3 days. And really the headline here is that whatever approach you take to give the intradermal injection, whether you've given them for 10 years or this is your first intradermal injection, it makes no difference. You get very high seroconversion rates, you get very high post-vaccination titers that you can see up here. So really the idea that intradermal injections are very difficult and if you don't do them properly you'll be wasting your time should be put to bed. And sort of based on this and other data, fractional doses of IPV are now recommended for both campaign use and also routine use by the WHO. And then considering our genetically stable OPV, so the novel OPV type 2, NOPV2, has been genetically engineered from the Sabin virus to prevent the genetic reversion events which are associated with vaccine-derived polio. So essentially it's more genetically stable. And because of the rapid increase in the outbreaks of vaccine-derived polio type 2, in 2018-19. This was the first vaccine globally, in fact, before the COVID vaccines to be given emergency use listing. So this is listing for use in emergencies before normal licensure processes. So we've gone on and done the key phase 3 licensure trial of this vaccine, recruiting 3,000 or just under 3,000 children over the course of around 6 months. Expecting the data from this very quickly, but broadly speaking, we've got very high immunogenicity data completeness, very high safety data completeness, obviously, which is essential if one is going to license a new vaccine. This trial led by Dr. Choge. So based on this, we expect NOPV2 to be licensed and pre-qualified, and it's likely, almost certain, to replace all other type 2 OPV going forward. So this would be the first new poliovirus vaccine licensed for decades. Now thinking about measles and rubella elimination, and this has been touched on again by Beata, and reducing zero-dose children. So some 50 years after the Gambia first demonstrated that it was possible to eliminate measles through vaccination, we've already seen various versions of this picture, but essentially the rates of coverage with measles vaccines are way below the 95% coverage you require to interrupt transmission, even with first dose here. And therefore we're a long way off elimination at the moment, particularly in sub-Saharan Africa, although other parts of the world as well. And many countries in this situation, because their population immunity is so low, they end up having to do outbreak response or preventative campaigns, so vaccinate all 9-month-olds to 5-year-olds over the course of a couple of weeks, which is very disruptive and very disruptive to normal services. So essentially strategies both to make campaign-based delivery of measles and rubella vaccines easier, as well as to facilitate increased coverage, are likely to be important if measles elimination particularly, but associated rubella elimination is going to be realistic over the decade. So we've been looking at this microneedle patch. So in this, essentially the measles and rubella in this case is embedded within these dissolvable microneedles. The patch is placed on the skin, the microneedles penetrate the skin and the microneedles dissolve, delivering the vaccine into the dermis. So this is potentially advantageous in the sense that the dermis has got more antigen presenting cells in it, so we may even find the immune responses are better. But the other advantage is it doesn't generate any sharps waste, so there's no risks of sharps injuries. It's easy to administer, so even in a campaign you could perhaps use Red Cross volunteers, for example, as they do in normal OPV campaigns. It's thermostable, so it's got decreased cold chain requirements, which is a really major burden on the EPI system here. And it's pain-free, which is obviously an important benefit for many parents. And the WHO have stated that microneedle patches have the potential to become game changers for achieving measles and rubella elimination. Distressed by having his patch on his arm. 1-2 double dummy AIDS de-escalation trial led by Dr. Adegwemi, who you can half see behind his mask there. So this is the first trial of microneedle patches in children anywhere globally, and it's also the first trial of measles and rubella microneedle patches. So we recruited the adults and the toddlers, had a data monitoring committee. This was obviously immunogenicity and safety data in January. It's focused on safety, of course. This is a phase 1 trial, but we're also collecting immunogenicity data, so the standard marker of immunogenicity for measles, neutralizing antibodies, T-cell responses. But we're also looking at acceptability. And we hope that this trial will generate the first data on what could be a potentially game-changing technology for future vaccine delivery, not necessarily just for measles and rubella. We were discussing hepatitis B only a couple of days ago as well. And lastly, just to touch on HPV vaccine introductions and life course vaccination. So the rates of cervical cancer mortality on the left and the progress with HPV vaccine introduction on the right could hardly illustrate global health inequality any more effectively, and likely also gender inequality. So it's a horrible image. So strategies to make HPV vaccine introduction easier, and also subsequently to monitor their impact on meaningful endpoints, is being led by Dr. Obiemi, who you can see in the corner. So first of all, we're looking at a reduced age indication for HPV vaccination. So HPV vaccines, as you know, are standardly given from 9 years and above. But what became clear during the initial delivery of HPV vaccines in the Gambia was that these is done through a school-based program predominantly, also of course aiming to catch out-of-school girls as well, of course, but it was focused on schools. And if you have a class with 7, 8 and 9-year-olds in one class and you're only vaccinating half a class at a time, this is really not a very efficient way of getting good coverage. So first of all, we're looking at the reduced age indication, and also looking at 1-dose versus 2-dose schedules. And anybody who's got any interest in HPV vaccines will know that's a very hot topic at the moment, and there's lots of exciting data and policy decisions on that being made already. So we are completing now follow-up of around 1,700 females in this study, looking at safety in the 4 to 8-year-olds by comparison to the older groups, of course, immunogenicity, and also Eunice presented some of her exploratory immunology that she's doing. We're also trying to explore parents' views of these vaccines. If you're giving a vaccine which is ultimately for a sexually transmitted infection, of course, how they will view that if this was given in a younger age group, and potentially, looking forward younger again. So the data on new schedules we hope will facilitate HPV vaccine introduction and improve coverage. And lastly, just to think about monitoring the subsequent impact of HPV vaccines as they're rolled out here. So HPV vaccines are, again, incredibly good vaccines. Their efficacy is exceptionally high, and based on data, randomized controlled trial data from high-income countries. But how that then translates into impact data in West Africa, where there may be a number of differences in terms of sexual mixing patterns, of course, some differences in HPV types, and a number of other factors which could impact on how well these vaccines are working. So Dr. Basharan will lead a baseline, so this is just at the point the HPV vaccines are being introduced, so a baseline age-stratified cluster survey to generate baseline data on HPV as well as other STI prevalence within the BASE HDSS. So we're aiming to recruit around 10,000 15 to 49-year-olds over the coming 12 months or exposures to allow us to monitor future vaccine impact over the coming 5, 10, 20 years going forward as obviously people get older. And I wasn't going to mention this, but as Chris mentioned it yesterday and encouraged me, this is not a trial, but it's something, a quite sort of interesting study that we got funding for between myself, Chris Murray and Umberto, about a month or so ago. So this is really to look at drones for transporting vaccines, one, potentially to improve vaccine timely hepatitis B birth dose in principle, and also to transport blood samples in vaccine trials. So this is a study we'll hopefully be getting off the ground. No deadly silence in the coming few weeks. So vaccine trials so I hope I've demonstrated that we've got a diverse programme of vaccine trials. Grant Mackenzie will expand on this later for sure, and Annette Earhart as well, Immunisation Agenda 2030. So i.e. to prevent disease, to promote equity, and to build strong programmes. And in the last slide, just looking forward, so Beata outlined many of the opportunities and challenges, which I'm not therefore going to start rehearsing in this one slide. So I just wanted to come very briefly back to the idea of vaccine equity or vaccine inequity, because I'm sure none of us are naive enough in reality, and the point has already been made, none of us are naive enough to believe that a fractional dose or a reduced dose schedule or even a magic microneedle patch are going to overcome problems of vaccine inequity in isolation. These are obviously very complex issues. There are numerous factors playing into whether a child is vaccinated. And in the same paper, you can see how challenging it becomes if one tries to think of a nice, clean intervention one is going to do to improve vaccine coverage in this setting and other settings, of course. Dr. Wareri had already presented, and Beata mentioned this. He's looking in his PhD, looking at some of these issues, so starting to try to dissect out some of these issues. And Ali Usau, who's the EPI data manager, is also doing really important work in this area. But I think it gives us an opportunity looking forward to consider the design and evaluation of more complex interventions. So in the future, we're not just confident that vaccines are safe and efficacious or safe and immunogenic, as we obviously find out from trials, but also that they're getting to the people who really need them, and therefore that the goals of the agenda are met and nobody is ultimately left behind. So thank you, without which, as everybody has said, we could certainly do none of this research. And thank you, assuming you were, and thank you for listening.
Thank you very much. We have a time constraint, but we will open the floor for questions. Yes. Can you move the mic, please? Can you help with the mic?
My name is Toyin Togun. I'm with the Clinical Research Department at the London School and a clinician scientist with the vaccines and immunotherapy. Given what is going on globally in terms of the unequal access to COVID-19 vaccine and monkeypox, which has been appropriately termed as vaccines apartheid, there's now a growing call to increase to develop capacity and the capability to manufacture vaccines on the continent in Africa. And yesterday there was also a talk about improving capacity for genomic surveillance within health systems. From your experience, given that you interface with manufacturers, policymakers, and so on, how do you think we can capitalize on this momentum and the angst that are prevalent on the continent in Africa? That's the question.
Okay, how long have we got? And it's a question to which your colleague on the left will have at least as much to say as me. Reality that until monkeypox hit Europe and the United States, nobody was taking this disease particularly serious. We've all known about monkeypox in West Africa for a very long time, although I'd never seen a case. And the fact that there were vaccines potentially sitting on shelves, we had the same thing with Ebola. So I think there is also a sort of zeitgeist thing. Things do come together eventually. CEPI has played a very important role in this, and companies are being pulled up on saying, why have you not invested in this space? And to some degree, we need to probably come up with a model of vaccine financing that is different from what we're doing now. There's talk about an equivalent to the Global Fund for vaccines, and essentially if they are incentivized. And I think without incentivization, it's going to be very, very difficult. But I wonder if our colleague from WHO wants to add to the discussion.
Thanks. I probably was going to ask the same. I think it's really a valid point, and I know that Africa CDC, especially under Dr. Nkenga's song, as they've been trying to push for this. the need is there, but I think, as you say, we really need the model to make sure that it's not like every country will want to do this. And this is really, we have to be, I know it's politically correct, but we really have to step in a place where not every country starts to go into this market space because it is going to be challenging. So it's really more about regional manufacturing on the continent. And we've been having discussions with our colleagues at Africa CDC to make sure that really North Africa and not in West and not in South. I know South Africa is already in this space. So yes, valid, but we have to be strategic on the models that we really push. And since now, I was the incident manager during the large urban yellow fever outbreak in 2016 in Angola and the DRC. And at that time we did dosing, a fifth of it. The challenge was that actually we didn't embed within that studies, longitudinal studies to try to look at dosing to control outbreaks. I mean, if you don't have enough doses, yes, you can give, but then how the protective efficacy for the fractional dosing and for yellow fever, the one we know as lifelong immunity, we don't know how long the MRC might want to consider.
I mean, funnily enough, we've got funding actually from the MRC to look at a fractional dose yellow fever vaccine trial. In fact, ironically, we're just about in various stages of starting 3 different yellow fever vaccine trials, looking at boosting related to the point that you're concerned about with young children, but also a trial looking at fractional doses that was funded through the MRC, and also fractional doses and also co-administration. And one thing there is now some fractional dose data, as you know, and that's in the outbreak. The CDC generated data on the immunogenicity of the fractional doses in DRC, didn't they? But we're just starting a study of might be better than subcutaneous injections. I mean, it seems to be fairly arbitrary what route people choose. For IPV, historically, people have always used intradermal injections for fractional doses. For yellow fever, in the outbreak, they used subcutaneous doses, and obviously the immunogenicity was good, but then there's concerns about the duration. So yes, we are actually specifically just, again, early in the new year, we should be starting a trial looking at fractional dose yellow fever as well, yeah, looking at some of those questions. So thank you.
Last question, yes? In the chat, but I don't have access to the chat. I don't know if, okay, can you the question?
Thank you very much. This is from Julia Mesikwa, Pat. She says, thank you, Ed and Prof. Beate for the excellent presentations. Are there any future plans to conduct vaccine trials in Uganda with the MRCG, since Uganda is a high burden? And a second question from the same person, Dr. Ed, following up from the presentation of Professor Kalibu.
Una pregunta rápida. ¿Te vas a conectar a la reunión esta que fue cancelada la semana pasada?
Sí, pues...
Can you please mute the person?
I can translate if anybody wants.
Oh, sorry about that. You can mute her. Okay, yes, it's clear. Sorry about that. Muted her. Okay. The second question is, Dr. Ed, following up from the presentation of Professor Kalibu on vaccine capacity at MRCG Uganda, how do you envision some vaccine studies in Uganda, such as the HPV, climate change, and others? Thank you.
I think Dr. Ledouari will actually, Professor Ledouari will actually refer to some of the vaccine studies that are currently going on in Uganda. So Julia was a PhD student of ours here at the unit in the vaccine field between the MRC unit in the Gambia and the MRC unit in Uganda. And there's certainly amazing strengths in Uganda, especially in the viral space. So when it comes to the surveillance, I was really delighted to see all of Pontiano's data on COVID. We just can do things better together, for sure.
Do you want to?
I think that addresses it. Yeah, I mean, Kirsty is doing a number of trials there, or Professor Ledouari, who'll be speaking later. So yes, there are surely opportunities that we're not currently taking, I guess.
Okay, very last question, because we have time constraint and have to conclude this session. Yeah, go ahead, please.
Okay. Thank you, Chair. I work mostly in the communities, and when you go into the community as for one moment, the people say, oh, you've come and taken our knowledge, and then you've gone. And I'm beginning to reflect, especially in these issues of vaccines, reflect on the final product. Are we looking forward at a time when there's supposed to be a partnership between the users of the product, that is the industry, and some benefit coming back to those communities that participated in those trials in terms of economic benefit? Because it seems as if after the research, everything goes on to the industry, and then the communities that promote benefit for their own promotion. Are we reflecting on this? Thank you.
That's a big and challenging question. Yeah, I mean, I think we as a unit are embedded within communities across the Gambia, so we are not bringing in an industry trial and then disappearing. We are working with communities, building capacity in communities. Many of the participants in our studies, receive care. Successful in the trials that we do because we provide good feedback to the communities so that they understand and they're very much a part of the trial itself. When we start a new community, I mean, we've started trials in a number of new areas where we haven't had quite such an interaction, and it has taken us some time to sort of build up that interaction so that we do get that level of community support. And I think the other thing that's critical at the end of the day, many of these trials have resulted in vaccine implementation through the government. So that's perhaps Grant can speak to this extensively related to the pneumococcal studies where there's a lot of support provided, but also, that's been dramatic in terms of the impact of doing those early trials in the Gambia in terms of then rolling out the programs in the Gambia. So obviously, with new vaccines, we can't promise if this vaccine is successful, you'll be the first people to get it. That's just not realistic, and that would be, incentivization as well. We're not trying to buy people to join trial. Often people are coming to our health centres to say, have you got a trial going on? People come, people bring their children, people bring their children's children, sort of thing. So we, in some senses in the Gambia, because we've been here so long, we've got this established relationship, hopefully.
Thank you. I think the discussion can be continued during the coffee break. You can approach the two presenters if you still have questions. This will conclude our session, and I would like really to thank the two presenters. Thank you very much.
Good morning. My name is Dr. Momluti Yansi. I'm the Deputy Director of Health Services, Ministry of Health, stepping in for the Director of Health Services, Dr. Mustafa Bitte, who is unavoidably absent due to other engagement. So I'm the chair for this 1 hour section, so I'm going to be very strict with time. Each presenter is given 20 minutes, and you have to make sure you stick to the 20 minutes given to you. So the first presenter, unfortunately, she couldn't make it to this event due to circumstances beyond. And the first presenter is Professor UK Research and Innovation Feature Leader Fellow and Associate Professor of Vaccinology and Immunology, working between MRC, Uganda Virus Research Institute at London School of Hygiene and Tropical Medicine, Uganda, Makerere University, and St. George University of London. So her presentation is centered on future maternal immunization. So it's a recorded presentation, so the control room, can we please play the recorded
Good morning.
presentation.
My name is Kirsty Ledware, and I'm going to be talking to you about the future of maternal immunisation. I'm very sad that I'm not able to be with you in person to give this talk, but I'm actually at a maternal immunisation working group meeting in Seattle. I hope that in the next 15 minutes I'll be able to give you a little bit of background to maternal vaccination and where we go next. So first of all, what is the problem we're trying to solve with maternal vaccination? Well, although the under 5 mortality rate has declined year on year and has never been so low, the proportion of deaths in neonates has not declined by the same amount. And in fact, in 2020, of the 5 million deaths in under 5s, approximately half of these occurred in the neonatal period. And even if you're able to survive an insult in the first month of life, then 2.2 million survivors go on to have neurodevelopmental impairment. So moving past survival and into thriving still remains a huge problem. Well, how can maternal vaccination help this? Well, maternal vaccination works by reducing this window of vulnerability here on the left as the grey bars, which is the time at which the antibodies that mothers pass in their placenta to babies reduces after birth. And at the same time, the infant's own immune system is still trying to catch up, which leaves a gap in the immunity when babies are particularly susceptible to infection. What we try with maternal immunisation is shift that blue curve to the right. So on the right-hand side, you will see a hypothetical scenario where antibodies have been boosted through maternal immunisation such that that window of vulnerability has really reduced, protecting infants in those vulnerable first few months of life. And indeed, maternal immunisation itself isn't particularly new. The first maternal immunisation actually occurred against smallpox in 1879, and you'll see in this timeline that subsequent initiatives such as tetanus, maternal tetanus immunisation first in the 40s and then in the 60s, followed by landmarks in regulatory approvals to include women in clinical trials in the early 1980s, followed by the WHO's maternal tetanus immunisation initiative, have really changed the landscape of maternal immunisation in the latter part of the last century. And since the 1990s, over 130 million women have been vaccinated against tetanus to prevent neonatal disease. And at the same time, other vaccines have been tested in pregnant women, including the Gambian trial of Haemophilus influenzae in pregnancy and the Gambia's participation in the RSV vaccine trials, which are hopefully soon coming to market. And at the same time, maternal vaccine clinical trials have changed. So as the timeline moved from tetanus toxoid in the 1960s to maternal influenza in 2010, followed quickly by maternal pertussis in 2012, following the epidemic of pertussis, and then in 2021, COVID-19 vaccine, followed by GBS and RSV, which are in development. And in 2005, all of the clinical trials for vaccines occurred in high-income countries. However, that changed 10 years later when at least 25% of these vaccines came from countries in Africa. So South Africa and Uganda were included in vaccine trials against maternal group B streptococcus, in addition to the USA, Europe and the United Kingdom. So what are the landmarks of maternal immunisation? Well, first let's talk about tetanus. WHO recommended tetanus immunisation in pregnancy, 2 doses in the first pregnancy, followed by 1 dose up to a maximum of 5 doses in subsequent pregnancies. And here in the chart you can see the line, which is the incidence of neonatal tetanus, following the cases of tetanus year by year since implementation of maternal vaccine studies, maternal vaccines vaccine against tetanus, and here you can see how that drops off a cliff as the coverage of maternal immunisation against tetanus rises. So a huge benefit for maternal vaccine study. 50% vaccine efficacy when the vaccines were first introduced into the UK, and most recently the FDA approval in October specifically for third trimester use to prevent whooping cough in the first 2 months of life showed a 78% efficacy in preventing pertussis disease in infants under 2 years of age when administered in the third trimester without any safety signals for fetus and the newborn. So again, another landmark ruling in terms of bringing safe and effective vaccines to prevent diseases in pregnant women and their infants. And what about other vaccines? So I mentioned influenza vaccine, and again, influenza vaccine benefits both maternal and child in proven influenza in infants with a 29% reduction in any respiratory illness and 42% reduction in clinic visits from this trial done in the US. In pregnant women, there was a 36% reduction of respiratory illness with fever and fewer clinic visits. So clearly, there's a huge number of protective benefits. In pregnant women was quite slow. And then along came COVID-19. And this is the COVID vaccine tracker developed by the London School and Ed Parker, who I hope is in the audience. And you'll see that as of last week, there were hundreds of vaccines in preclinical trial and over 20 licensed vaccines in use using a variety of different platforms, including novel platforms that have never before been tested in pregnant women or indeed in non-pregnant populations, including the mRNA vaccine, which is now so widely used. And why is this important? Well, because the COVID-19 vaccine programme was a potential game changer for maternal vaccines, both in terms of the speed in which the vaccines were developed, but also because of the advocacy that has gone on to include women in vaccine clinical trials and safety assessments to make sure that this data can be used for this pandemic and for all future pandemics, and getting pregnant women the vaccines that they need to protect them and their infants far sooner. And what's been shown with over 20,000 women in the US through the VSafe registry is that the vaccines are safe and effective. And there are now many studies, over 29 studies in 8 countries with over 300,000 participants that have not shown any safety signals from any of the vaccine platforms currently in use in pregnant women. And the WHO currently recommends 9 different vaccines for use in pregnancy. And this hopefully will facilitate other future vaccines, which I'm going to talk about now. So going back to the problem that we're trying to solve, deaths in the neonatal period are due to numerous different causes, but approximately 1/4 of all deaths are due to infections. And here you can see the global distribution of deaths amongst children under 5, with yellow being during the neonatal period, and the 2 largest ones there being sepsis and meningitis, and then other infections. And the top cause of infections in the neonatal period is actually Group B streptococcus. And it's estimated that a Group B streptococcal vaccine might prevent over 300,000 cases, 90,000 deaths and 57,000 stillbirths. And this is a great figure that Anna Seal developed for a modelling study looking at the global burden of GBS disease. And you can see that over 21 million pregnant women are colonised with GBS, which is the prerequisite for early onset disease and one of the major contributors to late onset disease. There are over 30,000 cases of maternal invasive GBS disease and 57,000 stillbirths, 320,000 cases of sepsis and meningitis in the first 3 months of life. And as I said, this is the leading cause of neonatal sepsis and meningitis. And there is also approximately 3.5 million preterm births that might be attributed to GBS. And currently, only the infant sepsis and meningitis cases and deaths would be covered by current programs to prevent group B streptococcus, whereas a potential vaccine would cover all of those areas highlighted in yellow, so clearly of benefit to both mother and baby. However, the road to GBS vaccine has been long and hard and is 50 years and counting, and this is mainly due to the enormous sample size that would be needed for a phase 3 trial. And even at an instance of 1 per thousand, 2 per thousand cases, such as in South Africa, a sample size of 40 to 60,000 women would be needed to be able to demonstrate efficacy. And that was considered to be totally unfeasible prior to COVID, and even since COVID this is a hugely costly undertaking. So different approaches required. One of those approaches is the Gaston consortium, of which Beata Kampmann and I are part, which aims to try and develop standardised assays in order to move operators from industry, from academia, from regulators and from funders to develop these assays. And these assays are then going to be used for a series of studies in the UK, in the US, in South Africa, in Uganda, to try and determine the serocorrelates of protection against invasive GBS disease through natural protection, whilst at the same time phase 1 and 2 studies in South Africa and Uganda are underway, shortly followed by the phase 3 trials, which are due to start next year. And this should then lead to one of 3 different pathways for licensure of these vaccines. The first is through a standard disease endpoints clinical trial, and the other 2 would offer immunological correlates for licensure followed by an effectiveness study. And I think option 2 here, which is a phase 3 study with accelerated licensure based on a serocorrelate, is the most likely outcome for GBS. And then let's think about RSV, where the information is also hotting up for an RSV vaccine, and this may well be the first maternal vaccine to market. And we know that RSV is an important cause of viral lower respiratory tract infection in young infants, with 34 million annual cases in children under the age of 5 versus 14 million for pneumococcal pneumonia and 8 million for Haemophilus influenzae. And 3.4 million children develop serious lower respiratory tract infection and need hospitalisation. And here you can see that between 1997 and 2006, in that 0 to 2 months of age, that's where the highest burden of hospitalisation is per 1,000 children. So clearly this is at a time when vaccinating the child themselves would be too early, and so we need maternal immunisation to protect these vulnerable infants. And even more so than GBS, you can see here the pipeline of vaccines for the RSV vaccine, which are in different stages of development, all the way through from preclinical and up to phase 2. And really excitingly, Pfizer on the 1st of November announced positive top-line data from their phase 3 maternal vaccine trial, which indicated over 80% efficacy for severe medically attended RSV in the first 90 days of life, and over 69.4% efficacy in infants at a later stage of medically attended RSV disease. So, in summary, maternal immunisation is a rapid birth up to those first 6 months of life, and there has been steady progress in acceptability of maternal vaccine programs over the last 5 years, with maternal vaccination with earlier inclusion of pregnant and lactating women in clinical trials. And there are 2 key vaccines, Group B streptococcus and RSV, designed for administration in pregnancy on the horizon. But there are still many unanswered questions. So what are the next steps for maternal immunisation? Well, we still need to improve equity of access, because maternal vaccines are still more likely to happen in high than low resource settings. We need more data on safety, especially of the newer platforms such as the mRNA vaccines, and we need them in higher burden settings, which require strengthening of capacity for safety assessments. We need to work with the communities in which we work to build confidence of maternal vaccines. And this is the double-edged sword that we've seen through COVID-19. And we need to consider vaccine schedules. If we're now vaccinating pregnant women, should this be considered the first vaccination in pregnancy, and should this be considered the infant's first dose? And do we therefore need to alter the infant vaccination schedules? And we also need to consider how these platforms could be rapidly utilised to prevent severe and serious infections from new and emerging pathogens, for example, Ebola, which is top of mind here in Uganda at the moment, or the Zika virus. And obviously this is a hugely collaborative piece of work with teams far and wide, but I'd like to thank our funders and my core team here on one of our community outreach programmes in Uganda. Thank you very much.
So if anyone in the audience and those who have joined virtually, if you have any question for the presenter, you can direct your question to Professor Kampman, who is here. She will answer the question on behalf of the presenter. Thank you. So the next presenter is Dr. Toyin Tugo. He is an associate professor at the London School of Hygiene and Tropical Medicine and a clinician scientist in the vaccine and immunity team of the MRC unit, the Gambia. So he's going to present on childhood TB in West Africa. Thank you. Over to you.
Good. So we're just waiting for the slides to be projected.
Don't worry, I will keep your time for you.
You have to deduct all that one.
Sure.
Okay, thank you very much. And my name is Toyin, and I'm delighted to present this talk on the growing portfolio of interdisciplinary research on childhood tuberculosis that we are conducting within the collaborative partnership of the MRC unit, the Gambia, in West Africa. And I would like to start with full disclosures. The data that I will be presenting the data that I'll be presenting are from research studies funded by the listed agencies, and the opinions that I'll express in my presentation are solely mine and do not necessarily represent those of the funders. For the scope of my work, I will first talk about the childhood TB, our work within the childhood TB work package of the West African Network of Excellence for TB, AIDS and Malaria, and that will be followed by a quick overview of about the newly established platform for research on post-TB lung health in children and adolescents in West Africa. Now, the WANITAM, that's the West African Network of Excellence for TB, AIDS, and Malaria, is an EDCTP-funded research capacity building network comprising 25 research institutions across 12 West African countries, supported by 5 European partners. The TB network within the WANITAM consortium is coordinated through the MRC unit in the Gambia, and it consists of 2 main work packages: the drug-resistant TB work package that is led by Professor Martin Antonio, and the childhood TB work package that I am leading. The strategic aim of the childhood TB work package is to improve the referral, diagnosis, and management of childhood tuberculosis through an integrated program of research and training on drug-sensitive and drug-resistant tuberculosis within national health systems in West Africa. There's a couple of trainings that we organized a couple of years ago. To the left of your screen was a training on diagnosis, management, and management of childhood tuberculosis, and to come up with a to get an overview of the most important or the main challenging research questions about pediatric tuberculosis. This took place at the Jos University Teaching Hospital in north-central Nigeria in June 2019 and was attended by about 30 healthcare workers comprising pediatricians, general physicians, and representatives of national TB programs from about 12 West African countries. This training resulted in the formation of the West African Pediatric TB Network, which is now recognized as a sub-network within the TB network of WANETAM. And to the right was another training that we organized at the Noguchi Memorial Institute of Medical Research in Accra, Ghana, focusing more on the laboratory scientists. And the aim was to train these efforts of tuberculosis in adults and in children, as well as to utilize samples for diagnosis of extrapulmonary tuberculosis. So following this series of trainings between 2019 and 2021, we launched a project to pragmatically investigate the diagnostic accuracy of GeneXpert Ultra Assay for diagnosis of tuberculosis in children within national health systems. And we recruited more than 200 children with presumptive TB, were recruited and investigated under routine clinical and laboratory conditions at the in three countries which are part of our network: the National Teaching Hospital for Tuberculosis and Respiratory Diseases in Cotonou, Benin, Kolebu Teaching Hospital, Accra, Ghana, and the Gabriel Touré Teaching Hospital in Bamako, Mali. We also used this study to establish the capacity for electronic data collection through internet-enabled tablets and the transfer of the electronic data by synchronization to a centralized database that is hosted at the MRC unit in the Gambia. This project was integrated into the overall WANETAM research career development objective, and the postdoctoral fellow was competitively identified from within the network, and they underwent a program of research training at the MRC unit, which I'll mention later. Now, the manuscript reporting this work is currently being finalized for submission, and we're looking forward to presenting the data at the next EDCTP Biennial Forum. But the figure to the left of your slide is showing one of the main findings from this project, in which we use Bayesian random effect meta-analysis to account for the heterogeneity between the countries to estimate the pooled sensitivity and specificity of GeneXpert Ultra Assay to diagnose tuberculosis in children using routine respiratory samples and using culture as the reference standard. The sensitivity was 5% and the specificity was 95%. This reported sensitivity is almost 15 to 20% lower than the reported sensitivity of GeneXpert Ultra from relatively well-controlled studies, as well as systematic reviews and meta-analysis. And the implication of this is that if we are going to end tuberculosis in 2030, 2035, while we need new diagnostics, it's also important that the currently available diagnostic tools must be optimally utilized, because this is a finding from a pragmatic study within 3 main hospitals across West Africa. To the right of your slide is Dr. Awaba Diallo. She was the postdoctoral fellow that worked under my supervision, who is leading the manuscript writing. She completed the research training fellowship, and she wants a couple of follow-on fellowships, and I'm happy to announce that she's now been appointed as an assistant professor at the Sheikh Anta Diop University in Dakar, now adding to the growing pool of female early career scientists that we're growing within the network in West Africa. Building on the work from the previous WANETAM project, and we renewed funding from the EDCTP. We are now about to start another childhood TB project that's going to take on for the next 3 or 4 years. And the aim now is to investigate the added value of non-sputum-based approaches for diagnosis of tuberculosis in children. And specifically, we will be investigating the utility of computer-aided detection of tuberculosis that provides automated interpretation of chest radiographs to determine the likelihood of tuberculosis in children through the application of artificial intelligence to chest x-rays. And we also investigate the added value of testing stool samples with GeneXpert Ultra to improve case detection of tuberculosis. We're no longer working on Fujilam because of some issues with the manufacturing, the kits that are currently available. The approach is that we're going to consecutively recruit children with presumptive TB at these 3 of our WANETAM TB partner sites in Cotonou, Kolebu, Accra, Ghana, and here at the MRC unit in the Gambia. And this is going to be an interdisciplinary project, because there will be a qualitative component where we aim to explore the feasibility and the end user acceptability of these non-sputum-based approaches for diagnosis of TB in children. And again, we are integrating this research into the overall one-time research career development objective. And in this regard, 2 additional childhood TB trainees have now joined my team. To the left is Dr. Vitrup, and to the right is Dr. Sheila Owusu, a trainee pediatrician from the University for Developmental Studies in Tamale, Ghana, who's now started a PhD, registered at the ITD Faculty of Infectious and Tropical Diseases at the LSHTM, and she will be based here in the Gambia for research on post-TB disability in children and adult post-TB disability. To give a quick background, treated for tuberculosis have persistent health impairment and relatively higher mobility and mortality when compared with the general population, but published data about the long-term impact of pulmonary tuberculosis on the lung function in children and adolescents, especially in TB endemic settings where the burden of TB is high. But I can say that is until now. This is a newly published data from the work of my PhD student, Dr. Esin Inkerewen, who is jointly supervised by myself. Treatment in children and adolescents is strongly associated with significant impairment in lung function and reduced health-related quality of life. Specifically, we show that Gambian children who were successfully treated for tuberculosis at least 6 months previously had almost 4 times relatively higher odds of lung function impairment, predominantly of the restrictive type, when compared with age-matched comparison group of children who were recruited from within the same households, but who have never been, especially in African children. So with this established capacity for pediatric spirometry and for research on a platform for clinical, epidemiological, and applied health research on post-TB lung disease in children and adolescents, and our partners included Professor Jimmy Whitworth asked me on Tuesday that are we having challenges to continue our research on during the pandemic, and I said yes, of course, but that the challenges provided an opportunity to be innovative. This is one of the ways in which we turned the challenge into an innovation. Given the delays occasioned by the COVID-19 pandemic and restrictions to travel, I worked with the Pan-African Thoracic Society and led the development of the first online course on pediatric spirometry. This training took the foundational spirometry course of the Pan-African Thoracic Society to the next level by incorporating multimedia components in English and French with demonstration video. So we broadened the scope of those that could attend the course to include both healthcare workers from Anglophone and Francophone countries. And more than 50 healthcare workers from about 10 West African countries registered for the course, and 98% of them successfully completed the course, and they used that to start accumulating credits for accreditation by the Pan-African Thoracic Society. We also used the trade credits to competitively identify some healthcare workers who were now sponsored and invited to the MRC unit in the Gambia for an intensive in-person training on pediatric spirometry that took place in October 2021. About 20 healthcare workers from 6 West African countries attended this training that included theoretical and practical sessions, including calibration of the spirometer, as well as practical session on performing spirometry in children and adolescents. And with the funds from this grant, we also donated brand new spirometers to 2 of our main partner institutions, Jos University Teaching Hospital in Nigeria, as well as Kolebu Teaching Hospital in Accra, Ghana. With this virtual and in-person training, we've established a network of committed pediatric lung health investigators with the capacity to perform lung function measurement in children and adolescents. Now, this project was an interdisciplinary one, as I mentioned, so we also organized an international multi-stakeholder workshop on post-TB disability in West Africa. It was a 3-day in-person workshop that took place in the Gambia with 40 participants. Half of them were women from 6 West African countries, and it included TB survivors, representatives of advocacy organizations, as well as policymakers from the WHO, national TB programs, as well as the Stop TB Partnership. We embedded a qualitative study within this participatory workshop, and the primary data collection was by in-depth interviews as well as a facilitator-guided focus group discussions. And using the deductive thematic approach for the qualitative data analysis, 3 main themes emerged from the lived experiences of the TB survivors. First is that they continue to experience reduced quality of life, affecting several aspects of their family and work life, as well as social participation. Secondly, despite completion of TB treatment, they continue to experience TB-associated stigma, and there's also the need for psychosocial support. Policymakers, on the other hand, emphasize the fact that there's knowledge gap about post-TB disability and that there's still inadequate data to support policy adoption. They also emphasize the fact that the current TB care package is limited because care for TB patients stops at the end of TB treatment, and that there's a need for new recommendations and guidelines to aid healthcare workers in providing adequate management for TB patients beyond the end of their TB treatment. The full manuscript reporting this work has just been accepted for publication in the Public Health Action, which is the official journal of the World TB Union, and is going to be available open access. So with this program of activities, we've now established a multi-country and interdisciplinary platform of research, and we are for post-TB lung health, and we are now ideally positioned to be able to establish multi-country prospective cohorts where we could determine the prevalence or determinants and risk factors of post-TB disability, and integrate basic science studies looking at immunological drivers of post-TB disability in children and adolescents, as well as social science studies exploring the impact of post-TB disability on their overall psychosocial health. We've started experiencing some early successes. Some months ago, we've just been awarded a new seed funding by the UK Medical Research Foundation for a new project that will be starting in the Gambia, and this is looking at generating ethnically appropriate reference values for lung function measurement among Gambian children. The background to this is that they currently utilize Global Lung Initiative reference values that is used to interpret lung function measurements, whether for asthma, COPD in children and adolescents, comprises data from Caucasians, African Americans, and Asians, with not a single data from indigenous African populations. So we're hoping that with this study, we can update the Global Lung Initiative reference values and providing data that could be generalizable to the rest of West Africa and Africa to provide a more accurate and reliable interpretation of lung function measurement. Also, we've been invited to co-organize the second International Post-Tuberculosis Symposium that will take place in the first quarter of 2023 in Stellenbosch, South Africa. And at the ongoing World Conference on Lung Health, we have a couple of presentations, and tomorrow I will be chairing the session on life after TB at the World Conference. And I think I've come to the most important slide, which is an opportunity to acknowledge
Good. Thank you very much, Dr. Twain. So So the next presenter is Deborah Nyirenda. She is an associate group head for community engagement and bioethic at Malawi Liverpool Wellcome Trust and Global Health Bioethic Network Senior Fellow. She has over 10 years experience of working in multidisciplinary research projects. Thank you very much, Deborah. The floor is yours. So can we give them time for them to work on the presentation?
Maybe while we're waiting for them to sort out the presentation, I can take this time to introduce myself. I don't know if you're able to hear me at the back.
Yes, your voice is loud and clear.
Okay. Can you hear me now? Okay, thank you. As he has already introduced me, my name is Deborah Nyirenda, and I'm from the Malawi Liverpool Wellcome Trust in Blantyre. Malawi Liverpool Wellcome Trust is an affiliate to University of Malawi, Kamuzu University of Health Sciences, KUHES, and Liverpool School of Tropical Medicine. So I'm happy to say that MOW has also celebrated 25 years of conducting research in Malawi, but I wasn't part of the celebration, so I'm so privileged to be here and be part of the 75th anniversary at the Gambia unit. So thanks for inviting me here.
Is that day 3?
Yes, day 3, yes.
You upload? To begin with, I would like to start with a history of community engagement because it is important to understand the history for us to know where we should be going. So, community engagement has a long history in rural development, and the main purpose of community engagement was to empower communities to co-create solutions to their problems. And the reason why community engagement was being promoted in rural development was because communities have got better understanding of their issues, and we need their voice to come up with solutions. And by engaging them in co-creating solutions, it helps to ensure that the intervention is relevant to their needs, and it also promotes ownership of the intervention as well as sustainability. In global health research, community engagement became popular in the 1980s because people living with HIV and AIDS started demanding to be involved in HIV drug trials because they believed that their voice was very important in development of drugs. And the slogan that nothing about us without us became popular then because it was a way of advocating that if we as scientists want to improve health, we need to make sure that we incorporate the voices of affected communities in whatever we're trying to do so that
our work becomes relevant to their needs. So community engagement, you'll agree with me that it has now become a requirement by most funders, including NIHR, Welcome, and several others. So what is community engagement? Oftentimes, community engagement is confused with community sensitization meeting or just community meetings. But according to the National Institute of Healthcare and Excellence, community engagement is defined as a process of getting communities involved in decisions that affect them, including planning, development, management of services, and activities which aim to improve their health and reduce inequalities. I've highlighted the words planning, development, and management just to show that involvement of communities is supposed to happen throughout the research cycle and not just at the point where we want to recruit participants. There are three overlapping aims of community engagement. I've referred to them as overlapping because there is a thin line between these overlapping aims, which are namely to enhance ethical research practice, to empower communities, and to rebuild trust with communities. And I'm going to talk through each of these aims. So you'll agree with me that in low and middle income, very poor, health services are not that good, and in some settings there's illiteracy, high levels of illiteracy. So such factors is too high. What that means is that the most poor will not be able to access those bed nets. And it's only the middle class and the rich people who will be able to afford, thereby contributing to inequalities. So that is an injustice if participating research communities don't get to benefit from the interventions that we have developed from the research that we have conducted with them. So community engagement is meant to empower communities to have a say on the design and delivery of health interventions. It's meant to give us a platform to understand what would be the barriers for them to adopt that intervention, what would be the facilitators. And finally, the third aim of community engagement is to rebuild trust with communities. Research in several sub-Saharan African countries has shown that negative past events have contributed to mistrust between researchers and communities. For instance, I can give an example of polio vaccine in northern part of Nigeria in Kano. It was resisted by the communities because of historical experiences with a drug trial by Pfizer on meningitis. Because there were several deaths experienced among the children, that historical experience influenced those community members to start resisting polio vaccine. So, in addition to that, global health is also critiqued as being top-down and perpetuating academic imperialism. What I'm trying to say is that global health has been critiqued that it's top-down and it's usually those with power who determine what the research priorities should be and the research practices. So community engagement is promoted to help decolonize research by privileging the voices of the less privileged or by privileging voices of local people and in a way rebuild trust with communities. So there's been several frameworks that have been used to help us to think through the levels of engagement with communities, and I've just picked one, which is a very old publication, but it's been widely used in the literature on participatory governance. So what this ladder is trying to say is that the lowest levels of engagement is where you just get people together with an aim of providing therapy or just to educate them. And that is non-participation because you have not empowered them. In the middle rungs of the ladder, what it says is that if you just involve communities with an aim of just informing them or to consult them and not incorporate their views, then that's tokenistic engagement. It's not meaningful engagement. And higher levels of engagement involve establishing partnerships with communities, delegating power, and empowering them to lead with implementation of the interventions. So to sum up everything that I have just said, what this all means is that in practice, community engagement has got to start right from the start in priority setting, where communities are involved in setting priorities for research, they are involved in research design to make sure that the study procedures are acceptable in that community, they are able to give their perspectives in terms of their lived experiences with the interventions, they are involved with the analysis to make sure that it reflects their experiences and as well as in dissemination of findings. In our recent review, which hasn't been published, we've looked at a number of approaches that researchers have used community engagement in medical research, and the most common approach is the community advisory board, where selected community members are invited and they're supposed to represent community voices in research. Following this, a number of publications have also focused on community meetings where they'll gather people together, inform them about research or to consult them about research. And there's been a number of studies as well involving community volunteers as peer educators or as leads who should help to implement interventions. And there's been several publications as well, combining several community engagement approaches. I'll not focus so much on the research focus on this of these publications, but most of the publications that have involved community engagement have focused on the disease burdens in Africa, such as HIV and AIDS, malaria, TB, and Ebola. So at Malawi Liverpool Wellcome Trust, we've conducted a number of research on community engagement, and most of it has also been done in other Wellcome-funded sites in Africa and Asia. So to just quickly go through that busy slide, we've tried to explore ways of engaging with communities. At the far left, bottom left, we've tried to engage communities by using radio and having a phone-in program where they should have dialogue with researchers and ask questions directly to the researchers. We've also explored digital storytelling, where we have given communities cameras to take pictures and tell us the experiences of their involvement in research. We've tried focus group discussions to engage them in specific issues, and we've also taken a critical stance on community engagement. And these are just selected publications that we have worked on. So this morning, I just thought I should share my reflections on the work that we've done on community engagement. I may not be able to say everything, but yeah. So from our reflections, based on the work that we've done, we've learned that it is important to understand what we mean by community. So a community can be defined as a group of people residing in a specific geographical setting. It can be defined as a group of people sharing same characteristics, such as maybe teenage pregnant mothers, or it can be used to define a group of people who have got a similar disease condition. So we need to understand what we mean by communities, because it becomes pointless for us to just engage, for instance, a community leader if we won't engage the affected communities. So we need to have a specific community in mind that we want to engage and make sure that these are targeted. In addition to this, it's important that we have to understand the historical experiences that communities have had with the various interventions, because like I have already alluded to, their historical experiences is going to shape how they engage with research and even how they engage with researchers. Local beliefs about diseases are very important for us to take into consideration, because if we don't understand what their local beliefs are around that intervention, we end up wasting a lot of resources conducting research on things that will not value. While in most African settings, the leadership style that they use is autocratic. So we need to make sure that we understand those power structures and make sure that everyone involved understands what we want them to be. Otherwise, it ends up becoming tokenistic. We've also learned that it is important to understand communication approaches because the communities have got their own ways of engaging among themselves. Communities have got their own ways of engaging among themselves, so it is important that we have to align our communication approaches to the existing communication approaches in that particular community. So it's pointless, for instance, to say that you're going to use social media to talk about research in a rural setting in Malawi, where people don't even have access to smartphones or internet. And one of the things that we have to be mindful of is hypothetical acceptability. What I'm trying to say here is that after working in a particular setting for a long time, you develop relationships with communities to the extent that sometimes participation in that research is based on maybe their intention to access certain benefits from that institution. And that same intervention, if it's being rolled outside that trial setting, it becomes resisted by the communities. So what this means is that sometimes when we do a lot of community awareness, a lot of community sensitization, and we've come up with data that this intervention has worked, it may not work in another setting where there isn't so much community awareness. So we've got to be mindful of how our interactions with the communities for several years is going to shape how they respond to research and how that may not be the same experience in another setting, or how that finding may not be generalizable in another setting. We've also learned that communities rely on social networks for information. So oftentimes, safety of the interventions that we bring to them is validated through we are introducing measles vaccine. We go the next day and tell them we are here to improve your health. We are introducing polio vaccine. And then we go another day with a different kind of message with an aim of improving health. And we keep on going, and they don't see the benefit of what we're trying to do. It leads to multiple uncontested realities that we shouldn't just dismiss, but we just need to understand what is the message behind that rumor. So open and ongoing dialogue is very important for us to pick their concerns. Sorry, the slides to have an understanding of their concerns, even while we are implementing the interventions. And okay, I'll just continue. I'll just carry on. Yeah. So the last point that I wanted to make on the lessons that we've learned is that communities rely on social capital, meaning to say that they rely on social networks for support or for information and everything else. So their participation in research sometimes is based on this sort of understanding that you're going to reciprocate that sort of kindness. Like what someone else alluded to say that how do we ensure that the research that we do benefits community? So we've learned that oftentimes when they are enrolling in research, they've got expectations, they've got very high expectations of us that will really address their health problems and even beyond that. So we need to make sure that we understand their expectations and we are able to manage those expectations. We are able to justify what we are able to do and what we are not able to do. Because all this can have an impact on adoption of interventions that may potentially or that have got the potential to address some of the problems that they're having. So to conclude, I would like to end with a quote by Albert Einstein that everybody is a genius, but if you judge a fish by its ability to climb a tree, it will live its whole life believing that it is stupid. What I mean here is that as scientists, we've got biomedical knowledge. We've got this sort of expertise, but we need to respect communities' expertise as well. They've got better understanding of their social cultural context, and they've got better understanding of their local history. And if we make an effort to understand their local perspectives and incorporate their input in designing and delivering the interventions, will help to make sure that the interventions are aligned to their needs, and that beyond the trial, those interventions will be adopted by their communities, and will also help to minimize wastage of resources, because we won't just end up spending millions of pounds on trials that will not have any impact on research. It will ultimately help to improve health outcomes. Thank you so much, everyone, for your attention, and thanks to my funders and everyone. Yeah.
Thank you very much, Dr. Deborah, for that brilliant presentation. I apologize for the interruption of electricity on and off. So the floor now is open for questions, comments, and contribution. Yes. Can you please introduce yourself when you are asking question? Thank you.
We need the mic.
They need the mic.
Good morning. Thank you, Dr. Deborah, for this great presentation on community engagement. I am Amel Zemsi, the head of the clinical trials unit at MRCG. So I have two questions, if that is okay. The first one is on the community advisory board. On your opinion or experience, do you take researcher, and do you feel that the need of community advisory board is not actually the reflection of the failure of ethical committees? Because in fine, how different are they? So that's my first question. My second question is, do you have any practical tips for these researchers
we haven't quite explored the idea of coming up with an independent community advisory board, but that would be something worth exploring. But I'm also aware that there are other community advisory boards who function very well in other settings because they've been properly empowered to do it. So at the end of the day, it's how you frame your terms of references with them and how you... The second question was to do with how different is the community advisory board from an ethics review committee, right? So I would say that these are very different committees in the sense that an ethics research committee is tasked with the responsibility of reviewing your protocol and identify ethical issues, while a community advisory group goes further than that. They're supposed to give their perspective. Research is done. So this is how these two are different, yeah? And then your last question was to do with practical tips on early engagement. So there are several ways that you can go about this. You can do, for instance, formative focus group discussions, where you identify community priorities and make sure that these are taken into consideration. And when we talk about engagement, it doesn't necessarily mean that we are engaging with the same kind of individuals at all the levels of research. So if you think that they may not have the scientific expertise, for instance, to contribute towards the design, aren't there any other stakeholders in the Ministry of Health or at the health facility who can provide that sort of expertise? So it's just an issue of what question are you trying to ask, who can you engage, who would be the relevant person, and at the end of the day, the expertise that we're looking out for them is not the biomedical science expertise, it's their understanding of their socio-cultural context. That's why you must not judge them based on their ability to climb a tree.
Can I? Good morning.
Can go ahead. So if your mic is even in problem, you can go ahead.
Oh, okay. Yeah.
So you can go ahead.
Okay. Thank you very much. This is a comment from one of the online participants. Her name is Helen Nabuera. She says, another great talk. We all have a lot more to learn about how to engage the communities we work in on the design, implementation.
Okay, good morning, and thanks for the presenter for the great talk. My question is directed to Dr. Toyin Togun. I'm Dr. Leopold Chenchi. I'm an assistant professor at the London School and also a senior scientist here at the MRC unit, the Gambia. So I have two questions for you. The first one relates to the findings about the sensitivity of the Genespa Ultra that you presented. So practically, when you go in the field, the sensitivity is like 20% or lower than what is reported from research setting. I'd like you to expand a bit on maybe findings that explain that gap in terms of the sensitivity. And also, since your research is ongoing, how you are thinking about addressing those moving forward. So the second question is about the reference spirometry standard that you are trying to establish in the Gambia. I mean, you'll be aware that when we talk about normal individual in terms of exposure, air pollution is a great challenge. How are you going to address that, given that people, depending where they are living, and can have exposure that is far disastrous than what you anticipate when they are diseased? Thank you.
All right. Thank you very much. Can you hear me? Okay, for the first question about what might be the factors responsible for the relatively lower sensitivity of the expert ultra following the pragmatic evaluation. Now, one of the earliest things I learned when I started research from one of my mentors was that a good research is not determined by where it's published. A good research is a research that actually leads to a more important question. And I think we work Awaba Diallo is doing, which I'm overseeing. The fact is that for the relatively lower sensitivity of 100% and specificity of 100%, but if there are challenges within the L system, it will still not perform. So I can sit here and give you answers. Whatever I say will be speculative. As far as I'm concerned, the answer to that should be from empirical evidence, and we are thinking of writing a forward. Right now, I'm leading the development of approaches to explore the L system issues that might be responsible, not just in children, because this is an indication that we're within national health systems, we're under-diagnosing tuberculosis, even with the routinely available diagnostic tools, not just in children. I'm sure this will be by extension, will also involve adults. So we're going to utilize the expertise within the London School TB Center, which I'm directing to such as to bring that together with quantitative approaches so that we have a very robust evidence that within the broad perspective of an implementation research to answer that question within West Africa. West Africa is a unique area in terms of heterogeneity of the TB epidemiology challenges within. With regard to your second question about generating ethnically appropriate reference values, very important question, because we know, from the talks that we've heard about climate change and planetary health, issues about both indoor air pollution and even pollutions in the environment. We have a series of inclusion and exclusion criteria that we will utilize. We're going to use we're going to recruit school-aged children. School-age children, basic school and, secondary schools, where we will have screening criteria to identify those children that will be recruited who will now have spirometry. So we're hoping to recruit that we hope, will include things like, ongoing chronic respiratory disease or acute respiratory infections. We also aim to, select schools based on the sampling framework within the Greater Banjul area that are relatively far from some of the, we know from available data, some of the heavily polluted in terms of environmental air pollution. So we'll most likely move away from those areas, possibly explore going up country where the degree of environmental pollution might be children, and which using robust statistical approaches generates what we think would be ethnically appropriate reference values that can be utilized for reliable and accurate interpretation of lung function measurement. Yeah.
Thank you very much. I entertain one more question, then we move. Doc, because we are going into the next session. So I think we entertain one more questions that we break for. Then.
My question actually goes to, I think, Dr. Deborah, right? I think community engagement is really important. I think journals nowadays actually ask for at what level have you actually engaged communities, even at the point of when you submit your research work for publication. What I struggle a bit with is when do we decide at what point should we engage communities, especially the advisory group that you've pointed out. In the MRC, for example, we go through steps really. If you have a research question, you develop that and go to the scientific coordinating committee before you move to the ethics committee that actually comprises of people that probably have scientific background and also lay people that you're supposed to draw from the communities. So really what I want to know is if you decide, for example, to go to the SCC and you get approval for, let's say, conducting a certain research, and you then decide to go back to the community advisory group and they said, well, this is not appropriate, maybe that is probably not the best decision or advice you'll probably get from them. How do you handle such situations, especially, for example, if you've tried to convince them that this is indeed supported by, evidence? Thank you.
Thank you again for the wonderful questions. So if I understood you properly, you're trying to find out when is the best time to engage with communities. Should it be at a time when you're about to submit a journal for a manuscript for publication, yeah? So I would say that according to CIOM's guidelines, engagement is supposed to happen at the earliest opportunity, meaning to say that before you even submit your protocol for ethics review, you're supposed to engage communities. Or even, maybe let me take it back a little. Before you even submit that concept to a funder, you're supposed to engage with communities. You're supposed to understand what their priorities, whether that sort of research will be acceptable, so that it's there in your grant application, as well as when you're submitting to ethics review committees. And I also talked about the principle of ongoing or sustained engagement, which is a platform where we could be able to identify unintended consequences of a particular research, for instance, and what do we do? I feel that if we really want to build relationships with these communities and we have identified a serious risk or harm that we can't resolve with communities, then we need to go back and seek amendments on our protocol. In the same ways that we do seek amendments for several other issues. Why shouldn't we, if we notice that communities are, for instance, facing problems with our intervention? Yeah? Priority setting as well, yeah?
Yeah, no, I'm just worried about, what is the best time to do this. Of course, this will differ, or it will vary depending on the research question. But if you're working in a community that is particularly difficult, for example, not receptive to research because of rumors and things like that, my trouble is how do you manage situations like that? But I think certainly the explanation you offer is helpful.
Break for wider explanation so we can give him a chance to ask his questions. Then we go to Dr. Ed. Thank you.
To the first presentation, and yet maternal immunization is really a great, not be doing these trials in phase 2 and phase 3, and it's really the issues around safety. So how robots are developed, especially which have been given to pregnant mothers, are safe. And I raise this because for COVID-19, really, the vaccines were developed very, very fast, and they got emergency use listing. But if you look at the safety monitoring system, passive spontaneous reporting is really not giving us all the data that we would like to do. And maybe a cohort event monitoring would help us to do that. In Ndola, for example, they are setting up long COVID, outpatient clinics, the same to Ghana, which will look at not only the sequelae of COVID, but also issues around safety of vaccine. So is that the future of this, to make sure that we are really certain that these vaccines are really safe? Because the numbers that we get from phase 2 and phase 3 trials will not be enough for us from a safety perspective.
This is such an important, and we need to think about a way that we're combining notification of events that might be occurring in the immunization system with the antenatal care provider side of things. And this is precisely a project, and Dr. Nyasi, I'm very happy you're here, that we have some funding for in the context of the COVID vaccines. We are currently recruiting 5,000 women on a passive surveillance system from various antenatal clinics in the country to follow up who did and who didn't get their COVID vaccination, to look for specific outcomes like premature birth, stillbirth, any issues with congenital abnormalities in the children, nothing to be expected there, but we need to document it. And by having a study that passively follows the women, it's important to have a data system that then feeds in with the government. Capacity to collect this kind of data and then transfer that crucial. And as part of that, and people who've worked with maternal immunization, to look at adjudication of events that might occur, because as much as Dr. Ledware presented the safety data, the majority of safety data are from mRNA vaccines. We have used a whole range of vaccines. We're at the brink of introducing 2 new maternal vaccines into the system. We do need to strengthen that regulatory framework, and we're working between EPI and MCA. A lot of progress needs to happen here, and the WHO Safety Advisory Monitoring Board, as you know, where I'm also an advisor, is working towards that. And it's really important to pull together the EPI side, the MCA side, and the perinatal care. So there's lots more work to be done, but to do it safely, we absolutely have to go there.
Thank you very much, Prof. So we'll take the last question, then we have a break. Thank you.
Thank you. It's just back to the community advisory boards and the participation, because I mean, we're trying to set, we're thinking about this actively related to the HPV prevalence survey that we're doing, which is going potentially, where clearly we need to do this effectively, without doubt. You sounded a bit skeptical about the community advisory boards, and maybe I misread you, but you sounded a bit skeptical. I mean, it's an easy way of doing it in some senses, but it risks tokenism maybe. What methodology would you advise? I mean, what are the best ways to avoid tokenism and get a genuinely valuable input from the community, I suppose, methodologically? Do you know what I mean? I mean, it's not an easy question. We could discuss it further, but.
Yeah, so maybe let me just try to highlight that. The reason why I sounded skeptical is based on our experiences, that our initial CAB, we had to dismiss it and come up with a new CAB. So that's why, I would say that I've got both positive and negative experiences of engaging them. But throughout the process, we've learned in terms of how best we can engage them. And we've also learned the importance of not just setting up the community advisory board, but ensuring that we've got a feedback mechanism where we are able to understand what the intended purpose of engaging them. So I would say that, to answer your question, I would say that community engagement or community advisory boards, they can effectively represent community interest if you have empowered the advisory board. So you can think of the different stakeholder groups that you want to reach out to and make sure that there's representation. So like in our setting, a number of institutions have tried to explore different approaches, like others have used residents in a particular setting. It's got its own limitations in terms of getting people based on where they reside, but they're getting them based on their social roles. So they would have a religious leader, a political community. So there are various models that you can follow, and yeah, we're happy to have a chat over this. Yeah.
Thank you very much. I think you can engage her for wider discussion during the coffee break. So my task has ended, so I will give the floor back to Prof. Hassan to take over. Thank you.
Thank you very much. Thank you. Deborah, very proud. Thanks so much. Thanks very much. Certainly, we won't deny your coffee because I know you need it after yesterday's wonderful dancing that was done. So we'll let you go and have a 30 minutes break and a coffee. So let's be back by 5 minutes to 12 for the next session.